Yeah, I can hear you now. We can hear you.
Good evening.
Yeah, and good morning to you.
Thank you.
Yeah. Just a few minutes and then we'll be starting.
Okay. Okay.
Yeah.
First waiting to admit the registration.
Good evening, Dr. Reddy. Connecting. Hello, good evening, Dr. Reddy.
Good evening, good evening, good evening.
Yeah, good evening. Thanks very much for joining us. Possible. Dr. Mandira?
Yes,
Hello?
sir.
Yeah, I think we can start. I think we can start.
All right. All right. Good evening. On behalf of Navrachna University, a warm welcome to all honorable dignitaries, guests, faculty, students, and delegates. The International Conference on Organic, Medicinal, and Pharmaceutical Chemistry, titled ICOM 2022, has been organized by the School of Science, Navrachna University. The conference will bring together international researchers, scientists, and specialists in various research and development fields of organic, medicinal, and pharmaceutical chemistry to discuss, debate, and exchange ideas in these fields and propose possible solutions to global challenges. It gives me immense pleasure on behalf of the conference team to invite you all to be a part of this inaugural function. Before the formal proceedings are embarked upon, I invite Navrachna University faculty to render a prayer and invoke blessings of the Almighty. I request you all to please rise for this invocation.
या कुन्दे मृतुषारहारधवला या शुप्रभास्तामृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवै सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाव्यापः
शुक्लां ब्रह्मविचारसार परमा आद्या जगद्व्यापिनीं वीणां पुस्तकधारिणीमभयदां जाड्यान्धकारापहां हस्ते स्फटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदां बुद्धिप्रदां शारदां बुद्धिप्रदां शारदाम्.
Thank you. I invite Professor A.V. Ramachandran, mentor, School of Science, Navrachna University, and convener of this international conference, to welcome this august audience. Over to you, sir.
Thank you, Dr. Mandira. A very good evening to one and all. Esteemed dignitaries of this inaugural session, our chief guest, Dr. D.S. Reddy, Director, IIM Jammu. Our keynote speaker, Professor M.S. Patel, our Provost of Navrachna University, Professor Pratyush Shankar, our Registrar, Navrachna University, Dr. Sandeep Vasant, my colleagues, plenary and invited speakers, participants, students, and invited speakers. It is indeed an honor and pleasure for me to welcome you all at this International Conference on Organic, Medicinal, and Pharmaceutical Chemistry, ICOM 2022. This is the first conference in chemistry being organized by the School of Science, Navrachna University. Previously, our school had organized 2 international conferences in the field of life science and 1 on public health on digital platform in line with the social distancing norms due to COVID-19 pandemic. Medicinal chemistry has taken quantum leaps in the past 3 decades, notably with developments in 3D structure analysis and understanding, high throughput screening, and virtual screening. The number of approved drugs for the market is decreasing in recent years and calls for more effective methods to discover a higher number of successful compound candidates. Medicinal chemistry being at the forefront is challenged here in this context, and also since it is the most time-consuming step in the drug discovery process. In the last decade, methodologies have been largely replaced from an individual scheme to an interdisciplinary team-oriented approach. Here, novel computational tools speeded up drug development such that promising lead compounds could be produced. Other new approaches were helpful, example, the omics approaches in modern life sciences that include genomic, transcriptomic, proteomic, and metabolomic information with reference bases that need to adapt computational, chemo, and bioinformatic tools. The sequencing of the whole human genome and the genomes of many other organisms has provided several new genetic and evolutionary insights and targets of pharmaceutical sciences. Drug discovery depends on the contribution from areas like in silico modeling, cheminformatics, and bioinformatics. Medicinal scientists are as yet mainly considering how to synthesize rather than what to synthesize in order to cure a particular disease. A transition from a trial and error approach to a predictive approach by new ways of targeted drug design has to be done that considers the metabolic functioning of human cell targets. In the future, medicinal chemistry has to turn even more to chemical biology, where molecular events are studied as pathophysiological processes. Drugs have to be designed toward the molecular targets of the disease cells and tissues. We have to obtain knowledge about the molecular base of metabolism, pharmacokinetics, and about the physiological effect of compounds. Promising future drug discovery will only be possible by the understanding and technological transfer of the molecular principles of drugs with their underlying metabolic transitions, pharmacokinetics, and toxicology. Organic chemistry is endowed with the tools to synthesize privileged structures called drugs, but the transition from drug to medicine requires appropriate methods of formulation and delivery and final journey through a regulatory procedure to be eligible for marketing. I'm sure that the next 3 days will provide a platform for integrating the 3 vital domains of organic, medicinal, and pharmaceutical chemistry to the practitioners in the field, and also provide a great learning experience for students of chemistry, life science, and biomedical science. With this note, I sign off, and once again, I extend a hearty welcome to one and all, and wish you all have a great academic experience. Thank you.
Thank you, Professor Ramchandran, for setting the tone for the international conference. I now invite Dr. Denny Mammon, Assistant Professor, School of Science, Navrachna University, to share details about ICOM 2022.
Good evening, dignitaries and all the participants of this inaugural function. Earlier developments in science took place when people worked in their niche areas of expertise. However, major breakthroughs in science have taken place when crossovers took place and focus in research shifted from individual disciplines to a more interdisciplinary and multidisciplinary approach. Major areas of this conference will be the various concepts and principles of techniques of organic synthesis, analysis, method validation, characterization, pharmacological and pharmaceutical evaluation of the organic compounds, in addition to metabolism, interactions between drug and receptors, and drug targeting. School of Science, Navrachna University has proudly hosted 3 international conferences. The first one on endocrinology and reproductive biology, the second on ecology and environment sustainability, and the third one on public health. We are proud to host the 4th international conference on organic, medicinal, and pharmaceutical chemistry. This conference will comprise of 8 speakers from abroad, as well as 21 speakers from India. Universities and institutes that are participating in this conference are University at Buffalo, the State University of New York, the Johns Hopkins School of Medicine, Baltimore, USA, University of Bordeaux, France, University of Avignon, France, University of Oxford, UK, University of Texas at Arlington, USA, Cornell University, USA, the Maharaja Sayajirao University of Baroda, University of Delhi, SBNIT Surat, Banaras Hindu University, Varanasi. Premier institutes within India such as JNCASR, ICER Pune, AIMS New Delhi, CSIR NCL Lab, CSIR CSMCRI, IIT Bombay, IIT Guwahati, and BITS Pilani. As also, we have contributions from major corporate giants such as Sun Pharmaceuticals, Cadila Healthcare, IPTA Laboratories, and Sood Chemi India Private Limited. More than 210 delegates have registered for the conference, with 52 research paper publications in both oral as well as poster modes to be presented over the 3 days of the conference. We hope that this will be an enriching experience to all the students, academicians, and industry professionals attending this conference. Thank you very much.
Thank you, Dr. Mammon, for sharing details about ICOM 2022. I now invite Professor Pratyush Shankar, Officiating Provost of Navrachna University and Dean, School of Environmental Design and Architecture, to address the audience. Over to you, sir.
Thank you, Mandira. And a very warm welcome to everyone present here. Welcome, Dr. Reddy. A very warm welcome to Dr. Patel, and to all the dignitaries, faculty members, heads of schools who have joined in an online mode. This conference was being planned to be offline, but unfortunately, as the times are such, it is in online mode. So I would like to welcome you all to Navrachna University. Before I say a few things about the conference, I think I would just like to give a perspective on Navrachna University for the benefit of everyone present here. And perhaps you would also understand why this conference absolutely aligns with the mandate and the ideological moorings of the university. This university is a relatively young university. We are around 12 years old. And from the very beginning, the university has the very foundational moments of the university was that we want to be a university that believes in broad-based education, that believes in interdisciplinary education, and that prepares a student to go out there to solve societal problems. In that sense, the conference, the theme of the conference is absolutely apt and on the line of what the university really wants its schools to be doing and its students to be doing. In our university, we focus a lot on students doing social projects, learning by engaging with the community. In our curriculum, in the recent times, we have integrated a lot of modules where they're actually working on real problems of the society, and in the process, becoming much, much more responsible citizens. I'm reminded of this quote that the real purpose of the university is to find something that is still not known. In that sense, the work that the School of Science under the able leadership of Dr. Ramchandran, the work that they have been doing is very, very important and very inspiring for all other schools of the university and all our faculty members and students. I am very impressed, and I really like to follow the work that they have been doing both in research and teaching. For one, in today's day and age where so much talk and so much insistence is on professional courses like MBBS and engineering, we really appreciate the work of School of Science to keep the interest alive in the subject of science, in the subject of pure science, and how they have managed to bring the excitement, the passion, and the possibilities of pure sciences or sciences at large to a very young audience. And to my mind, that is a very, very important contribution of the school. And I would really like to congratulate Dr. Ramchandran and your team here who have been consistently upholding the values of research. And in that sense, this conference is just an indication of that passion that you have all brought on board. With that, I would like to, sorry, there's my dog here. I would like to end my note. I would like to again congratulate the conference organizing team, and I would wish well to all the delegates. I hope they have a great time. Thank you.
Thank you, sir. That was Professor Pratyush Shankar, Officiating Provost, Navrachna University. It is indeed an honor to have in our midst today as chief guest, Dr. D.S. Reddy. Please allow me to introduce our esteemed chief guest. Dr. Srinivasa Reddy is Director, CSIR, Indian Institute of Integrative Medicine, Jammu, and CSIR, Central Drug Research Institute, Lucknow. He did his PhD from the University of Hyderabad in the year 2000 and his postdoctoral studies in the University of Chicago and University of Kansas, USA. Dr. Reddy has over 20 years of research experience post-PhD in total synthesis of natural products, medicinal chemistry, and drug discovery. He has 7 years of experience in the pharmaceutical industry, Dr. Reddy's and Tata Advinus. A molecule discovered by his team at industry is currently in human phase 2 clinical trials. Dr. Reddy is the author of 120 publications and an inventor with 35 patents to his credit. Dr. Reddy has won several awards, namely the J.C. Bose National Fellowship by SERB, Department of Science and Technology, Government of India, the Shanti Swarup Bhatnagar Prize in Chemical Sciences, the Nasi Reliance Industries Platinum Jubilee Award in the field of Physical Sciences, the Sun Pharma Research Award in the field of Pharmaceutical Sciences, the Chemical Research Society of India Bronze Medal in Chemical Sciences, and the Central Drug Research Institute Award for Excellence in Drug Discovery and Research in Chemical Sciences. Dr. Reddy is Fellow of the Indian Academy of Sciences and also Fellow of the National Academy of Sciences, India. He is editor of Bioorganic and Medicinal Chemistry Letters and Elsevier Journal. Sir, we welcome you to this international conference and invite you to address the audience.
Thank you. Thank you, ma'am. Can you hear me?
Yes, sir, very clearly.
Respected dignitaries, faculty members, and students. Good evening, good morning, depending on where you are. First of all, let me thank you for making me part of this international conference on organic, medicinal, and pharmaceutical chemistry. The chosen topic is very, very close to my heart, and all my research, all my experience is relevant to this particular topic, both organic, medicinal, and pharmaceutical chemistry. All years I am spending on this particular area. So I may be a little biased, but I strongly believe that people who do not know organic chemistry or medicinal chemistry, they are missing something in their life. At least to me, the kind of joy or satisfaction I get when I see on clean NMR, when students show me, or when I was working in during my PhD and postdoc times, or when you complete, when you synthesize natural product, the final molecule, the kind of satisfaction, joy you get, I don't think you can get any other place. I think that's where I feel that people who doesn't know about or who are not practicing organic chemistry, something they are missing in their life. Yeah. As you know, organic chemistry plays a role in everyone's lives and touches almost every aspect of our existence in something or other way. The role played by organic chemists in pharmaceutical industry continues to be one of the main drivers in the drug discovery process. You all know that one. Organic molecules perform functions in nature, drug, technology, and it plays as an engine for understanding the structure and reactivity. Although it plays a role in several aspects of life, considering today's conference topic purpose, let's focus on drugs. So I don't think we can imagine our life, our healthy life in particularly without medicines, what we, it's like part of our life, everyday's life. So how organic chemistry plays a role in this medicines or indirectly our daily life. So Indian pharmaceutical industry contributes substantially, not just for Indian, but also for the global healthcare. So some numbers to tell you for the students and for some of the faculty members, it supplies about 50 to 60% of vaccines. I'm sure you all know about recent vaccines. We all, because of COVID, we all know about how much we contributed in terms of vaccines, and 40% of generics medicines consumed in US, and about 25% of all the medicines in UK. Just I'm giving you some number. These are all available in the statistics from Indian pharmaceutical industry. So this is it's a big contribution to human lives from Indian pharmaceutical industry. So Indian drugs are exported to more than 200 countries in the world, with US being just being the key market. Of course, major share is coming from the US market. And generic drugs are they account almost 20% of global export in terms of volume, making the country's largest provider for generic medicines globally. And just for some more information, currently India has the highest number of US FDA approved plants, which are outside the US. Of course, in US you have much more, but when you look at other countries outside US, India has the maximum. And the Indian pharmaceutical industry is on the high growth, like more than at least numbers are available about 10 to 12%, like a huge opportunities. It is all possible because India has been doing extremely well in terms of quality of chemistry and quality of chemists we have in this country, particularly organic chemists in India. And we contributed so much, and I see there are so many opportunities in terms of novel technologies. There are new technologies are coming up, like for example, like continuous flow chemistry, reagent-less chemistry, and may help in addressing some of these environmental issues in the future. And I'm sure we are already, many Indian industry also already adapting to that. On the other side, Indian pharmaceutical industry has suffered external shocks, some time ago, Beijing Olympics in 2008, when many polluting industries are suddenly shut down in China. And also there was some issue with the Doklam standoff in 2017, if you remember. And recently also during the time of a pandemic in last couple of years, at least in early 2000, we really had a bad situation. Particularly there was a fear that China may stop supplying medicines, or some problem comes between us, then how are you going to survive? So that's where already government and also many industry leaders, they thought about it. Then they thought it's a big opportunity for us, both the policymakers and pharma industries, to carry out and retrospect past successes and explore ways to regain our global leadership in particularly in Atmanirbhar or Make in India way. I think there are several issues were taken up, and I'm sure chemists are people like us going to play a major role in addressing some of those issues. I thought just I'll tell you a couple of I'll show a couple of slides on drug discovery part. Can I share my slide? The way.
Yes, so you can share.
Can you see?
Yes.
Okay. I'm sure this slide, I'm sure many of you are aware, particularly who know drug discovery or who talk about medicinal chemistry, right? Just for the benefit of students. So any drug discovery process starts from target identification, then target validation, then there is something called hit, like initial hit, then for the lead optimization, and further optimization lead to candidate selection. Then once you identify preclinical candidate, it goes to like 3 different phases of human clinical trials, like a phase 1, phase 2, phase 3. Then of course, then you apply to regulatory authorities like DCGI or or FDA, US FDA, like European medical agencies, you apply to them and they give permission to market drug. So the cost is huge. It's associated, the cost more than $2 billion US, and time takes like 10 to 15 years. Like look at that and how much patience we need to, how much effort we need to put. And if you look at just numbers based on several studies, chemists like us makes about 10,000 compounds. Only 10% of them, that is about 1,000 compounds, get into pharmacology testing and safety testing. Out of that, only 10 of them can go to human clinical trials. That's not the market. That's only we'll get a chance to test in humans. So there's only 10 compounds. Out of 10 compounds, you get only one of them will get into the market. That's where you get into the medical shops, right? That means you look at this one statistics. Chemists like us makes about 10,000. Only one of the 10,000 compounds get a chance to hit into a medical shop or into the market. So that's kind of statistics are available, right? So this drug discovery is a very, very challenging field of science, and whole drug discovery process. I look at early drug discovery. Basically, identification of clinical candidate is going to play. That is going to be a very challenging. At the same time, we need to do very, very careful assessment there. So that's where we're going to determine our next success in clinical trials. So look at this one, like NCEs means like new chemical entities. When chemist makes, it will go through different stages like primary assays, then cell-based assays, then selectivity, different receptor like selectivity assays. Then once you are through with this one, then you go to in vitro admit assays, then in vivo models. There are also then a couple of models like acute models and also chronic models. Again, depending on disease, then we have to choose that models, advanced models, and finally exploratory tox criteria in different species. And finally, you identify a developmental candidate. So look at that funnel. Like basically, you'll be filtering compounds when you starting from compounds, NCEs, to go to the development candidates. So why I'm showing this particular slide is, so during this process, wherever you fail, wherever some problem comes, you need to go back to drawing board and design a new molecule and again synthesize, right? Synthesis of analogs, wherever, like in biochemical assay, you fail, or in selectivity assays or functional assays, or in PK, pharmacokinetics or toxicology, wherever you fail, you have to design again new molecule and again you go through this cycle. So all this cycle again start from organic chemistry. That's where you make molecule, you design molecule, make molecule. That's where I'm trying to convey you people is organic chemistry is going to play a major role, and particularly in discovering drugs and designed, synthesized by medicinal chemists. Many of them are originally trained as a organic chemist, and developing skills in organic chemistry, particularly students who want to discover drugs, is very, very important. That's what I wanted to convey this message. Just to a couple of slides, just to tell you a little bit about my institute, where I am presently heading this institute. This is CSIR, Indian Institute of Integrative Medicine. And this is one of the 37 laboratories under the umbrella of Council of Scientific and Industrial Research, that is CSIR. So our, like what we would like to is, like we'd like to have a center of excellence in natural product chemistry and biotechnology driven drug discovery, integrating modern biology with chemistry. That's what we aim for in this institute. We have two campuses. One is at Jammu, where presently I am staying, and we have branch in Srinagar. And in addition to that, we do have experimental farms like Gulmarg, Pulwama, Chata, Anantnag, and Leh. So they're basically medicinal and aromatic plants are cultivated, and we create value also for some of the products what we get there. And what we do overall core in our lab, so that is major focuses on natural product based drug discovery. We start in terms of medicinal chemistry, starting from hit to lead to preclinical candidate. And we have biotechnology, we have extensive research going on in biotechnology of plants and microbial and secondary metabolites. So fermentation-based technologies, again, we have another concept called phytopharmaceutical drugs. This is one of the new route, like NCE route, phytopharmaceutical drugs, which is based from plants. So this is also one of the area we are actively involved, like starting from good agricultural practices, good laboratory practices, good manufacturing practices, and finally we try to do good clinical practices. And we have a extensive plant sciences division. So we work on this enhancing the rural bioeconomy through cultivation. We have like enable accredited QC drugs and food essential oils. These are all some of the activities we do in our laboratory. Coming back to my own research group, of course, recently I moved here. All these years, last about 9-10 years, I was working in NCL Pune, National Chemical Laboratory. But recently, about 1.5 years back, I moved to this place, Indian Institute of Integrative Medicine. So what we do in our research group, I am a chemist. I'm an organic chemist by training. Of course, I did work in industry as a medicinal chemist, but in my research group, we do both total synthesis and medicinal chemistry. And we pick up molecule based on their biological activity and convert them into like hits and leads, and we'll take it forward for further biology, further processing towards inventing drugs. In addition to that, so overall, my research work in my group is application-oriented organic synthesis, and we collaborate extensively. Particularly look at this one, like a crop protection, insect repellents, and medicinal chemistry. These are all highly collaborative in nature, right? And what is the output we get? We get like a trained manpower, PhD students, postdocs, project assistants, summer interns, and that's what we like good human resources also we produce from our research group. And I looked at your program. I think excellent choice of speakers, and okay, very senior professor or very established professors like J.S. Yadav, Professor Bedekar, Fernandez, and several people from industry and outside, also different areas from industry like Dr. Rajmanar. I think you had a great like selection of speakers. Again, I would like to congratulate you all, and I wish you all the best for your conference. And I would like to once again congratulate, and I wish you all the best. And if you people need any particular help or any collaboration or anything, I'll be happy to connect with appropriate science. Thank you.
Thank you very much, Dr. Reddy.
Yep.
Thank you, Dr. Reddy. Thank you for a very informative session. Going ahead with the proceedings, the themes of the conference are organized under 3 broad categories: organic chemistry, medicinal chemistry, pharmaceutical chemistry. And the total number of papers that will be presented are 52. The book of abstracts that will now be released gives a brief summary of the papers that will be presented over the next 3 days. We will now have the release of the book of abstracts. Over to you, Dr. Parth Pandya. That is a glimpse of the book of abstracts for the International Conference on Organic, Medicinal, and Pharmaceutical Chemistry. Thank you. We are fortunate to have with us Professor M.S. Patel, who will deliver the keynote address. It is a privilege to introduce our keynote speaker, Professor Mulchand Patel. Professor Mulchand S. Patel is Distinguished Professor at the State University of New York, Buffalo, USA, 2008 till date. He is also UB Distinguished Professor, State University of New York at Buffalo, Jacobs School of Medicine and Biomedical Sciences, 2004 till date. Professor Patel's research focus areas include endocrinology, inherited metabolic disorders, metabolism, molecular basis of disease, neurobiology, protein function and structure, and transgenic organisms. Professor Patel did his PhD at the University of Illinois, USA, and his postdoctoral studies at the Fels Research Institute, Temple University, School of Medicine, USA. Professor Patel has received several awards and honors, namely the SUNY Distinguished Professor Award in 2008, the Research and Scholarship Award by the Research Foundation in 2007, the Pfizer Professorship in Diabetes Grant Award in 2007, the Oxygen Club of California Award 2006, the Stockton Kimball Award in 2004, the Sustained Achievement Award for Outstanding Achievement in Scholarly Activities 2002, and the Fulbright Research Scholar Award in 1987. Professor Patel has more than 250 research publications in journals of international repute, 3 book chapters, and professional memberships of reputed societies such as Society for Inherited Metabolic Disorders, International Society for Neurochemistry, American Society for Biochemistry and Molecular Biology. Sir, we welcome you to deliver the keynote address. Over to you, sir.
Thank you. Thank you very much for kind introduction. Before I proceed, I want to thank you, Professor Ramchand, for invitations. This has been a pleasure to go back to Vadodara. I'm used to call it Baroda because this is what I left in mid-60s with, Baroda as my home for 3 years that I work at Alembics and at MSU, where I got my master's degree, and went to USA for further educations. So today I'm going to talk about some of the things that I learned in Vadodara some more than 50 years ago, and that has been a continued interest for me over the last 5 decades in terms of my own thesis research as well as my own independent research activity. So as I mentioned over here, over the many years, I have continued to have research interest in 2 parallel topics. I was trained as nutritional biochemist. And so as you can see here, I have kept my hands on both parts of the biological sciences. I do metabolic studies, which are, metabolic programming as it relates to maternal obesity and fetal programming that really causes some of the major diseases of today's type 2 diabetes and obesity, which are programmed right in utero even before birth because of the maternal obesity. And this has been my research interest for the last 30 years or so. The interest in regulation of pyruvate dehydrogenase complex from the metabolic viewpoints and more, laterally in this understanding of structure functions in normal and disease states. And that has been for nearly for last 5 decades. Today, I'm going to talk about 2 specific aspects of this particular complex. Its structure is important for organization of various components of the protein to carry out the a sequential reaction to convert pyruvate into acetyl-CoA. And because of the key enzymes in glucose metabolism, its regulation is the key things in terms of understanding the derangement in metabolic processes, and especially in glucose metabolism as it relates to diabetes and obesity, and some types of cancer that I'm going to talk about. I'm going to relate, because of this particular enzyme is so important, its regulation, not only natural regulation within the cells, but when that regulation is derailed, the involvement or use of small molecules organically synthesized or isolated from plant material, and how these molecules can really help in terms of getting back to normal regulation of pyruvate dehydrogenase complex. So here is the one major metabolic scheme for glucose metabolism. Glucose or carbohydrate in general carry out about 50% of our daily caloric intake, and some of the amino acid carbon skeleton also convert into the compound called pyruvate. So when glucose is metabolized in the cell in the cytosolic compartment via glycolytic pathway, glucose is converted to pyruvate. And then pyruvate is transported into mitochondria, and the first reaction where pyruvate is converted to acetyl-CoA is carried out this complex, I call it pyruvate dehydrogenase complex or PDC. And so this is the gateway of glucose or carbohydrate metabolism. Once 3-carbon compound pyruvate is converted to 2-carbon acetyl-CoA, there is no way to go back this carbon back into the glucose synthesis. So this is the one way, that is the process that occurs at the level of PDC, and hence this enzyme is very much in regulation for allowing the complete use of glucose for either oxidative metabolism or biosynthetic processes. So let's consider this enzyme as a thing. In most of the cases in the glycolytic pathway, each enzyme carries out one reaction, and that is carried out by one protein, what we call specific enzyme. But here in this particular complex, there are 3 catalytic reactions are carried out, but they are in a sequential manner by different proteins. But these 3 enzymes are put into a complex which is very tightly formed as well as regulated. So there are 3 enzymes. They have a very specific name, pyruvate dehydrogenase, which is the first component called PDH. There is enzyme 2, dihydrolipoamide transacetylase, and the third component is called dihydrolipoyl dehydrogenase. So these 3 catalytic components, and the E2 and E3 are interconnected by a binding protein that is put into the subcomplex of E2 and B3 E3 binding protein, to which E3 binds. So this sequential reaction 3 are pyruvate is converted to CO2, and 2-carbon fragment is transferred to E1, which then takes the lipoyl moiety from E2 and transfers the 2-carbon segment onto the lipoyl moiety. E2 carries out the second reaction that forms the acetyl-CoA and releases, which can be used for biosynthetic processes. And then the lipoyl moiety, which is fully reduced, is reoxidized for the second reaction by the component 3. So the overall 3 reactions are independent, but the product of each reaction is not really released into the cells, but transferred directly. So this is called channeling, very effective process for carrying out this process. And ultimately, NADH is formed in this particular process. Acetyl-CoA can be oxidized in the mitochondria through TCA cycle and respiratory chain to produce ATP, or it can be transported out in the cytoplasm as citrate, which can be reconverted into acetyl-CoA for the synthesis of fatty acids and cholesterol. So the importance of the glucose oxidation to form ATP or for biosynthetic processes is regulated by this particular complex. It's also important to see that these are the reactions that I just mentioned here, normal metabolic processes of carbohydrate. But when something goes wrong, diabetes, obesity, strokes, and even some types of cancers are also, are involved in PDC regulations. There are genetic defects like, PDC deficiency at birth, which is very devastating and causes, mental retardations, and overall, life is very shortened without any treatment. Lactic acidosis and even the poison can interfere with the functioning of the PDC and can lead to very complications. There are some other chronic diseases in which PDC is implicated to have some interference, not directly, but secondary effect on the PDC regulations, and those are also of some general interest. So let me give you just general outline about what this reaction is. I just gave you the reaction about pyruvate being converted to acetyl-CoA by the PDC. Now PDC, because of the important enzyme, it remains in the active form, as well as it is phosphorylated and converted into inactive form of phosphorylated form of PDC. And there is a way to dephosphorylate and convert back into active. So this active and inactive conversions are carried out by two specific groups of enzymes, pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase called PDP and PDK. And they themselves are regulated by the product of the reaction, as you can see here, acetyl-CoA and NADH, or the substrate itself, which is the pyruvate over here. So there is a very neat internal regulation in which this cycle goes into active and inactive form to actually metabolize things. In addition to, there are other, metabolic regulation that I will discuss in a few minutes. So let's consider the overall, number of proteins which are involved in this complex and how they are actually put together as a functional molecule. So there is E1 has two different protein gene products, and the overall structure of this particular component, the first component, is alpha-2, beta-2, two proteins of alpha and two beta subunits. E2 is only homomer alpha structure, and E3 binding protein is a similar protein to E2, as I will show you. That is also has one single protein, but the protein itself is in the dimeric form. In addition to that, there is a dimeric form of E3, and then there are PDK, or actually there are 4 isozymes, so that in just to regulate this enzyme, 4 kinases are working on this particular complex. And there are 2 specific PDP phosphatases, PDP. So overall, there are 9 protein or so that are put together in this particular complex. Now the arrangement in the complex is very unique and very interesting. This was work I was studying as a graduate student, how this particular complex was put together. And so as you can see here, the E2 and binding proteins are in the center of the complex. They make the core of the complex. About 40 proteins or molecules of E2 and 20 of BP are put together, and there is a central core which contains 60 proteins. Of these 2 particular proteins are the thing. There is still a controversy about this thing, whether these are 40 plus 20 or 48 plus 12, but that's the further interest for the students to, if they are interested, they can go into that particular aspect of study. 30 molecules of E1 bind to the periphery of this thing, and about 12 proteins of E3 also bound onto this particular central core. And the core is this E2 molecule as the inner core that it carries out the reactions, but the extended by, on the N-terminal region, there are 2 lipoyl binding domain and 1 subunit binding domain. And on E2, the subunit binding is the E1 that we talked about. These 30 proteins are binding on the E2 binding sites. The BP is very similar to E2. Actually, it's by the gene duplication has been created. And as you can see here, that it has an inner domain, but no function. This is the binding protein, no catalytic activity, because one specific histidine molecule has been replaced by another amino acid, and no catalysis can be taken place. But the binding domain has for the binding of E3, and there is one lipoyl domain instead of two. Let me get this pen things. I'm still working with my, okay. So here is the lipoyl domain that is being, for the binding of kinases and the PDP that we'll see later on. To this complex, in addition to this thing, there are PDKs, and there are 4 different PDKs. 1 to 3 molecules of PDK are bound to this molecule, and then they just walk all over to phosphorylate this molecule. And PDP, there are 2 different types of PDP, and there are 2 to 3 molecules. So in terms of this entire complex, when put together, the total mass is more than 9 million Dalton. It's just like a ribosome, even larger than ribosome, except that the ribosome also contains RNA. This does not have the RNA. So if ribosome RNA is taken out, the rest of the protein molecule is as big or even smaller than PDC. So you can see the functionality of this particular molecule is very complex. As is shown here, just here, the same molecule shown E2 with its catalytic domain, E1 binding domain, and the 2 lipoyl domain. And to this lipoyl domain, there are 4 PDKs isozyme. They bind in a very specific manner. And one of them also binds to the lipoyl domain of the E3 binding protein. And 2 PDP are also interacting with this thing. So in terms of their specificity is also important, which molecules of PDK are going to bind which domain of this particular 2 proteins and how they will be regulating. So here is the cartoon way of showing it. Here is the center core of containing E2 and the E3 binding protein of the C-terminal regions, and that makes 60 mer of the complex. Their N-terminal domains are extended outward because they are, have this linker molecule, so they are not very rigid and quite flexible. To this thing, E1 binds to the E2 side, E3 binds on the binding protein, and PDKs and PDP can bind to this extended lipoyl domain in various things. So this is a very large molecule that extends in terms of various binding regions. Number of these components have been crystallized. We have crystallized 2 structures, E1 and E3, as it bound to the E3 binding proteins. Part of the problem with having the crystal structure for the entire molecule is that this extended molecules do not allow the crystallization of E2 molecule alone, and hence the PDC in itself totally cannot be crystallized. But most of the other parts of the molecule, except this extended part of the E2 and E3, have been crystallized. And there's a great deal of information about that, which is very helpful in terms of understanding of the catalysis as well as its regulation for binding small molecules that I will talk a little later. So here is the one that in the collaboration with Dr. Sijak, we crystallized the E1 component. It is alpha 2, beta 2, has 2 active sites because of the this dimeric structure of the molecules where thiamine diphosphate is bound. These 2 sites are chemically equivalent but functionally interdependent. They do not work in isolation. So when one cycle starts in the first active site, halfway the second active site is activated and initiate the reaction. So when the one site stops, the second site cannot work independently. So these are called half site reactivity of this particular enzyme. Based on this structure, and I'd like to introduce now that because of this kinases, there are specific serine residues in the alpha subunit of the PDH or E1, which are subject to phosphorylation. So covalent modification is the way to regulate this particular enzyme. And there are three specific residues of serine called site 1, site 2, and site 3 are located on the alpha subunit. When they get phosphorylated, enzyme becomes inactive. So here is the one thing. In one molecule, there are two alpha subunits, and each alpha subunit has 3 sites. So total of 6 serine, 3 here and 3 here not shown. These are the one that will be phosphorylated. Now the understanding what has been observed is that out of this 6 serine, any one serine gets phosphorylated, enzyme gets fully inactivated. So the question is, why do we need 6 molecules here to regulate this one particular molecule? So that's the one aspect. The second aspect, before we go on to that thing, let me show you that these 3 serines are located on 2 loops. There is one phosphor loop A on which site 1 and site 2 are located, which are very near to the active site, as I will show you. And the site 3 is located on another loop, which is called loop B. So when the serine is unphosphorylated, this loop are organized in a way so they are ordered and they do not interfere in the active site channel. When they get phosphorylated, they get disordered and they get relocated in the active site channel, and hence, do not allow the catalytic function to take place. So the phosphorylation indirectly, moves the loop A and B into places where it then interferes with the catalytic mechanism. So those are the, indirectly way phosphorylation really changes the structure within the complex molecule or within the E1 component, and hence it interferes with the catalysis. And here is the active site region. This is where the coenzyme thiamine pyrophosphate is located in the E1 component, and the serine 264, which is the site 1, which is located nearby on the loop A system. When that gets phosphorylated, this molecule or the loop moves into this channel and hence does not allow the binding of the pyruvate onto the thiamine biphosphate. So this is very, the interaction of, phosphorylation of serine and relocation of this particular loop within the structure of A really interferes with the molecule, and hence one can design the molecule which may not allow this loop A system to move into this particular direction. That would be the one approach for interfering with this, phosphorylation regulation. So the question is this thing, that we, as I mentioned, that one phosphorylation site gets phosphorylated, the enzyme gets inactivated. So why do you need 3 sites in the first place? And to regulate or to phosphorylate these 3 sites, one PDK would be sufficient to phosphorylate, while there are 4 dedicated kinases. 4 specific enzymes are made and transported in the mitochondria just to regulate one enzyme, just to do phosphorylation. So the question, it's very logic. Why would system will be designed in such a complex way when the simple one serine and one kinase would be sufficient to regulate the particular enzyme? But since this enzyme is so important in the body function for energetics, it is regulated in a very specific way. And the first thing we ask questions, why do we need these 3 sites? And if these 3 sites are there, why do we need 4 kinases, and what the interaction or specificity of this enzyme are for these 3 specific sites? So we made various mutants. This is the wild type. All 3 serines are, in the proper places. We mutated in such a way so that only one serine was left in the active, process, while the remaining 2 serines were replaced by alanine. And then we made a mutant which there was no serine at all. So enzyme was always functional. There's no control whatsoever. And then we tested its position towards 4 kinases. And as you can see here, here is the specificity of these 3 sites for 4 kinases, how they work. PDK1 phosphorylates all 3 sites, and that's the only enzyme that can phosphorylate site 3. All other 3 PDKs can phosphorylate only site 1 and site 2. The rate of reaction is quite separate. PDK1, PDK2, and PDK3 have highest site for reactivity for site 1. Site 2 is higher for PDK3, and intermediate things are, for the site 2, and site 3 is only phosphorylated. So there is a specificity, and the rate of reactions are quite different for different kinases, and that allows one way to regulate this particular enzyme. The major way to regulate this enzyme is its distribution in different tissues. We do not want to regulate PDC in all tissue in the same fashion. If that was the case, diabetic people won't even survive for a few weeks or a few months. Look at here, PDK is located only major organs which utilize carbohydrate in the major source. PDK2 is the same way, and the PDK4 are regulated in a somewhat different way by hormonal regulation. PDK3 is measured into the testes and brain and lung things, but they are not present in the major glucose or metabolizing tissues, such as liver and heart and skeletal muscle, which is where large amount of glucose is being consumed. So you can see here the tissue specificity allows to regulate this enzyme in a very specific way. So when the glucose metabolism is deranged in the diabetic conditions, we would not have any interference in the brain metabolism of glucose. Otherwise, there would be very severe complications and short-lived phenomena. So going back to that again, this previous slide, these regulations of PDC by PDKs and PDP are very important way in terms of regulating carbon flux of glucose into various metabolic pathway in a tissue-specific way. So you want to regulate or modulate glucose metabolism in some tissues, but not in the other tissue. And that really gives you one specific way of normal glucose metabolism in different tissues in the body during the fed and fasting condition, as I will show you later on. So here is the thing, how this enzyme is regulated as it is now organized in a specific way within the PDC molecule. In good health, fed and fasting condition, as we eat for some time, but most of the time we do not eat, so that its conversion from active to inactive and back to active depends upon whether we are eating something and we are in the fed condition. When external carbohydrate is provided, that is considered fed conditions. Fasting is like overnight fast, when we don't eat anything. The glucose metabolism needs to be regulated for glucose homeostasis. When this normal cycle gets deranged due to some metabolic diseases, the interference causes derangement in glucose metabolism, which leads to type 2 diabetes or obesity, where insulin resistance is the cause of interference in glucose metabolism. And then there are specific diseases, such as, I'm going to give you some example of cancer, where you can see that this would be another way to see how the whole system is completely redesigned for regulation purposes. So here is the simple thing, what we go through on a 24-hour cycles. So we go through fed conditions when we eat meal that contains a large amount of carbohydrate. It enhances plasma insulin secretion. So level of plasma insulin goes up. The meal suppresses glucagon secretion by the pancreas. So the level in the plasma goes down. The overall molar ratio of insulin to glucagon increases, and the insulin drives the glucose metabolism. And the insulin controls number of these reactions. Specifically, it down-regulates PDK, so the PDC is less phosphorylated. It enhances PDP, phosphatase reaction. So PDC is dephosphorylated, enzyme gets active, and the glucose derived from the diet can get converted to pyruvate to acetyl-CoA. Energy is derived from glucose oxidation, and the extra acetyl-CoA molecules can be used for fatty acid synthesis and cholesterol synthesis. And if you eat too much carbohydrate and low fat, you can still synthesize a lot of fat and deposit. So the consumption of total carbohydrate really can lead to development of obesity as well, But this is through the activation of pyruvate, PDC reactions in the liver. In the skeletal muscles, same skill, but the muscle does not synthesize fatty acid, so it uses for energy purposes. The same thing happens in various other tissues depending upon their capacity. Brain can take glucose and convert to ATP and really maintain its metabolic and the neurological activity based on glucose oxidation. During the fasting state, there is no glucose coming from the dietary source, so insulin secretion goes down by the pancreas, and what happens is insulin level goes down, glucagon secretion goes up, and the molar ratio of insulin to glucagon increases, and hence the PDC gets activated through PDK activations, and at the same time, PDP is down-regulated. Enzyme gets nearly, largely phosphorylated and inactive, and glucose carbon can be then directed from pyruvate going back to glucose synthesis via gluconeogenesis. So conservation of pyruvate oxidation is curtailed during fasting conditions, and we depend on fatty acid oxidation to provide energy in the liver and skeletal muscle. However, in the brain, nothing happens. Brain remains the same thing. In the brain, PDC activity is nearly 80% always active, irrespective of what we do in 24-hour cycle. Whether we eat or we are in the fed or fasting condition, that doesn't really affect. So in terms of brain, PDC regulation in the brain is very much constant within under the physiological conditions. Otherwise, brain only depends on glucose oxidation. It cannot oxidize fatty acids. So if we try to manipulate this thing, and this is achieved by distribution of PDK and PDP in the brain cells. PDK activity is very low, and PDP activity is maintained. So the enzyme is always kept in active form to nearly 80% all the time, which is not the case in the liver. Liver can go low activity in the fasting condition, 10 or 20%, and when we eat, it goes to 5 or 60% activity. So it fluctuates quite a bit to allow more flux through the pathway. So that's our normal things as we go through daily routine of eating, maybe religious fasting if you go for fasting for 2-3 days, and Shravan Mas, if you eat only once a day, your metabolic system will be adjusted, but within this normal range. Then something goes wrong in diabetic conditions where there is no insulin secretion, type 1 diabetes or type 2 diabetes where insulin secretion is very low. Then the PDK is activated, PDP is inhibited because of the low insulin level, and the whole system can be, controls the PDC in the inactive form, and the glucose metabolism is impaired, and the hyperglycemia develops in the type 2 diabetic condition and also in type 1 diabetes condition. To understand what can be done, that in this case, if you, the main important enzyme in this PDK isoform is the PDK4, which is insulin regulated in terms of its transcriptional control. So if we create the PDK4 deficiency, then the diabetes can be reversed. The glucose metabolism can be, again, come to a near normal level, and insulin not required, but PDK can be indirectly, PDK4 isozyme can be, interfere in terms of its regulation, then the regulation can take place. So here it shows indirectly that if you interfere by making PDK4 deficiency, which is the reversal of the diabetic condition, then the flux through the PDK will continue. Instead of PDK4 deficiency, if you have a small molecule that can specifically inhibit PDK4, then it is the similar things that the reversal of type 2 diabetes and glucose metabolism can be increased. And this will also then enhance the phosphorylation of AMP kinase that I will talk a little later. When that gets phosphorylated, enzyme gets gets activated, and that enhances the activity of the phosphodiesterase that actually degrades cyclic AMP. So in diabetic conditions, increase in cyclic AMP level is the cause that really impairs the glucose metabolism. So if you, instead of cyclic AMP being converted to simple AMP, then its action to the pKa through enhancement of gluconeogenic process would be taken out, and there would be less glucose synthesis. So indirect way of getting the PDK through, one can actually reverse the diabetic condition. Similar thing is under high-fat diet. When people eat a high-fat diet, it interferes with the glucose metabolism at the level of PDC because it generates acetyl-CoA, and acetyl-CoA enhances the PDK activity. So when we eat really again, but much of that activity in the obesity is through PDK2, and if we can create PDK2 deficiency either by gene knockout, is what this slide showed, but if we can also interfere with the small molecule by inhibiting PDK2, then the whole process can be reversed and glucose metabolism can be increased. So there are ways of doing in terms of small molecule used to specifically regulate the PDK2 and PDK4 activity, which would be very useful in terms of controlling type 2 diabetes or what is called fatty acid liver under the high-fat diet. So these are the important things under pathophysiological conditions where interference with PDK in a very specific manner can be very helpful to normalize or near normalize glucose metabolism in the body. So here is the general summary of what I have said so far. That the PDC regulation under normal conditions works with this tissue-specific distribution of PDKs and PDP and their relative ratio, their isozyme-specific regulation by metabolites like acetyl-CoA, NADH, and various pyruvate and various things. Phosphorylation site specificity of PDKs. PDK have very specific affinity for 3 different sites. And so that affinity also plays an important role in regulations. And the things that I have not given specific information, but gene regulations, hormone regulate gene regulations, specifically PDKs and especially PDK4, which is regulated by hormone like insulin. Insulin down regulates PDKs in general, but specifically PDK4. So this 4 specific way one can have the PDC regulation under normal metabolic conditions. So now what happens in the abnormal conditions? And I want to give you one specific example about cancer. Different types of cancers have different way of regulating PDC. The one that is very widely known is what is known as aerobic glycolysis. That Otto Warburg, observed first, indicated that glucose metabolism to pyruvate and to CO2 was impaired in some cancers. Even though the oxygen was available, glucose was not metabolized, indicating that the pyruvate metabolism was somehow impaired, and much of the pyruvate was converted to lactate. So even though oxygen was available and the mitochondria were there, the glucose only went to pyruvate and to lactate. And so it is called aerobic glycolysis. The whole process is glycolytic pathway and very little oxidative metabolism. So even in the presence of air or oxygen, the process was only glycolytic in nature. And this is an important thing. And that was his conclusion at that time was the dysfunctional mitochondria are likely responsible for this aerobic glycolysis that he observed. much more work has been done since then. And of course, now we know that the aerobic glycolysis is result of hypoxia in certain cell types, where hypoxia-induced factor 1 alpha, which then, now we know that it interferes with gene expression of glycolytic enzyme that increases many glycolytic enzymes. And at the same time, it inhibits some of the enzymes in the mitochondria, and specifically, actually, it induces PDK1. So if PDK1 goes up, it phosphorylates PDC, and the PDC is inactivated. So the oxidative metabolism derived from the glucose carbon cannot take place, and hence the pyruvate is converted to lactate. That was one simple explanation of, so PDK1 and PDK3 can be also, modulate PDC activity by inhibiting this particular complex. So here is the one example that in certain type of cancer where hypoxia is the major reason, this factor HIF1 then regulates PDK by increasing the transcriptional activity of PDK1 and PDK3, which in turn then phosphorylate PDH, and the PDH is equivalent of PDC in terms of functionality, and this enzyme gets inhibited, and much of the glycolysis, diverts its pyruvate into lactate formation. That is the one thing. And because of this interference, this reactive oxygen species is formed in the mitochondria through the complex 1 to complex 4, and that can generate derangement in mitochondrial metabolism and cellular processes. That was a simple explanation. Much more work in the last 10, 15 years that has come out is very interesting and very novel. That normal process of glycolysis and oxidative metabolism occurs in normal cells. In the cancer cells, and especially in some type of cancer cells, oncogene tyrosine kinases are induced that actually regulate the carbon flux through the glycolysis as well as through PDC. So this tyrosine kinase phosphorylate specific enzyme, pyruvate kinase muscle type in the glycolytic pathway, and they also enhance the activity of PDKs by phosphorylating PDKs, which in turn become more active, and they phosphorylate PDC and inhibit the pyruvate metabolism. So this is the indirect, not direct way, but through the PDKs, the tyrosine kinases, due to the cancer cell profile, really re-enhances the inactivation of PDC in many different ways. And I'm going to give you some specific example. So here is the 293 serine, which is the site 1 phosphorylation. When that gets phosphorylated, enzyme gets inactivated. Here is the tyrosine, which is 301, in very near vicinity of the 293. When that gets phosphorylated, that inhibits PDH by blocking substrate pyruvate. So just the way I mentioned earlier, phosphorylation of serine or site 1, that loop A was moving into the active site and inhibiting. There is another way of doing it, that by phosphorylating tyrosine molecule, that molecule also moves into the active sites and inhibits PDC activity. So this is another independent mechanism in the cancer cells only, how to regulate PDC activity. As we go through here, not only that it does PDC phosphorylation through tyrosine phosphorylation inactivation. It actually tyrosine kinases phosphorylate PDK1 in 3 specific sites, and especially tyrosine 136. When that gets phosphorylated, it actually enhances its activity, and hence it can also phosphorylate. So it also phosphorylates PDK1 and increases PDK1 activity. At the same time, this tyrosine kinases phosphorylate PDP in several different positions. At least 5 different tyrosine in the PDP can be phosphorylated, but more specifically, molecule 94, when gets phosphorylated, enzyme gets inhibited. So tyrosine kinases do PDC inactivation by 3 different ways. They directly phosphorylate PDH in the tyrosine in the active site region and inactivate. In addition to that, enhance PDK activity by tyrosine phosphorylate. They inhibit PDP by tyrosine phosphorylate. So 3 different ways they are, putting in the same direction as shown over here. PDP gets phosphorylated and it gets, its binding to the E2 binding gets diminished, and hence the PDC activity goes down. And so it's very important to see how in the cancer cells, this one normal mechanism is now additionally 3 different ways it's regulating PDC activity by PDH in tyrosine phosphorylation and PDK and PDP phosphorylation on the tyrosine molecule, and giving the overall enhancement of inactivation process of PDC. So here is shown over here that by tyrosine kinases, they can directly phosphorylate PDH molecule, tyrosine 301 as I showed earlier. They phosphorylate PDK1 and enhance its activity by tyrosine phosphorylate. That phosphorylates even readily a serine phosphorylation site 1. And they phosphorylate PDP and hence downregulate. So by downregulating this thing, increasing this activity, they phosphorylate site 1 very readily. At the same time, independently, they also phosphorylate tyrosine. So these are all different ways of doing regulations. And as you can see here, in some cancer types, the PDK isoforms and specific isoforms are affected in terms of enhancing this PDC inactivation with a poor prognosis for the outcome of various different cancer types. So this is one way to do it, So inhibition of PDC activity by tyrosine phosphorylation of PDH, PDK1, and PDP explains aerobic glycolysis. But that's not the case, because in some other cancer types, PDC activity is increased. So the question is, how do you explain increase in PDC activity in certain other cancer types, which are not, going through aerobic glycolysis, but they have increased oxidative metabolism and they need more PDC activity for glucose metabolism. So how do we explain that, And the mechanism is another third mechanism in terms of PDC regulation. So here is our normal PDC regulation through PDK by inhibiting the site 1 as the major site for phosphorylation of serine and that thing. In the cancer thing, when there is energy stress, the AMP kinase gets activated. And when that gets phosphorylated, it phosphorylates 2 serine molecules on PDH, not those 3 that we know, site 1, site 2, and site 3. But in addition to that, there are some other serines, and the serine 314 gets phosphorylated first, which then allows the phosphorylation of serine 295 near site 2, no, near site 1, but this is a different serine. When this gets phosphorylated, it does not allow the phosphorylation of serine 293, which is site 1. So phosphorylation of another serine nearby actually protects site 1 from phosphorylation and hence not inactivation. So AMP kinase in certain cancer types gets activated, and through this different mechanism of serine phosphorylation in terms promotes the not phosphorylation of site 1. And that is shown over here, that once the serine, this particular serine gets phosphorylated, it inhibits the PDK's phosphorylation of site 1. And that allows, and this also allows the phosphorylation of this thing. And once this thing is phosphorylated, it does not allow phosphorylation of 295. So the site 1 and the nearby another serine, they are really interacting and protecting each other, in a very specific way. So this is the third mechanism, totally different. So in different cancer types, either you can have more PDC or less PDC by phosphorylation or not allowing phosphorylation of site 1, one can have a very integrated molecule. So here is the PDK or AMP kinase that actually allows increased TCA cycle activity and oxidative metabolism. So AMP kinase phosphorylates this process, and then the whole thing gets, in terms of increased PDC activity in non-phosphorylation form. And this is very important for, metabolic activity of several enzymes or in several different types of cancers. That is very important. So this is the third mechanism in the cancer type, which is, not normally seen in the normal cell functioning. So here comes what you are interested in. How do we use small molecules to interfere with PDK activities or even tyrosine kinase activity? And there have been number of attempts, and there is, a good deal of information, but the story is not yet complete. This dichloroacetate is an analog of pyruvate, and that's a very known inhibitor of PDKs. The problem with that thing is that this compound is toxic because it's not permeable to cells, and especially not into mitochondria where the PDKs are. So that, activity requires large amount of TC, and that has a neurotoxicity because it gets metabolized and interferes with normal metabolism. Large number of, molecules have been isolated and tested and even synthesized some of these things. And they can, bind in various specific sites in the PDKs. All the structures of PDC proteins have known except the E2, as I mentioned earlier. So one can do the docking things and see where these molecules are, docking on the different proteins and how they might, act in terms of binding of the substrate or the various other molecules within the cells of PDK. And one can, really design number of molecules. So one approach is to synthesis of small molecules and understanding their action at the PDK and PDP level. Here, as I mentioned here, DCA is not soluble. So some, attempt has been made to have two DCA molecule attached to a different molecule that actually enhances delivery of more DCA molecule within the cell. Here is the mito-DCA, where there are 3 molecules of DCA are attached, and this molecule is more permeable to mitochondria, and some studies have been done in the cells, but still more work needs to be done in vivo. And there are a large number of molecules have been, been shown to be inhibitor in a very specific way to the isozymes of PDK. Recently, we tested phenyl butyrate, which has been shown to inhibit PDKs in the cells or in vitro. We have an animal model for PDC deficiency. So we actually use this particular molecule and found some interesting, benefits in terms of giving phenyl butyrate to mouse model with PDC deficiency. So there are ways to do, and that there's still more work needs to be done in terms of this process. Well, the story is not complete yet. PDC regulation requires another way. One of the product, which is acetyl-CoA, can be used to acetylate PDC. Alpha subunit of PDC can be acetylated, and hence it allows or brings PDK to bind to this particular molecule in a more active way to phosphorylate, and hence the enzyme can be inactivated. The SIRTUIN3 can deacetylate enzyme and make it active. So in terms of covalent modification, phosphorylation of serine is one way to regulate PDC. Regulation of tyrosine becomes very important in the cancer cell types, and the acetylations, another modification of PDC, is also an important aspect. So there are very different ways, and some of these things have come within the last 10, 15 years. It's such a new way of understanding of how this PDC gets regulated. So here is the summary of what I have said. The regulation of PDC in the inactive form, when it gets phosphorylated or acetylated directly, and then it can be converted into active form. In some cancer, it can enhance, mitochondrial respiration. And when it's inactivated, glycolysis can be, enhanced aerobic glycolysis in the cancer cell types. So it's very important to see how PDK and the small molecule will play a central role in metabolism in different cell types, especially in the cancer, is very important. It's very hot area, and I think it's, more work needs to be done. And I think there is very promising way to use the designing small molecules, and is used in terms of regulation of both aerobic glycolysis metabolism as well as oxidative metabolism in different cell types. So in summary, PDC plays a central role in oxidation of glucose. PDC regulation is achieved by tissue-specific manner of phosphorylation, dephosphorylation involving PDKs and PDP isozymes. Interaction of PDK and PDP with E2 as well as site-specific phosphorylation of E1 adds to regulation of normal PDC regulation. Phosphorylation by tyrosine residues by oncogene kinases, is a novel control for PDC inhibition in some cancer type, while AMP-mediated serine phosphorylation modulates PDC and in some other cell types, which is totally different from the tyrosine phosphorylations. And acetylation of PDC is yet another mechanism for control of PDC regulations. And all these regulations of novel regulations and even the normal regulation can be modulated by use of small molecules under specific conditions. So I think this particular molecule provides a rich arena in terms of designing different molecules and regulating different PDKs as well as PDP to modulate PDC activity under normal as well as disease states. And anti-cancer drugs are, I think, very promising field for young people who are, at the level of students or postdoctoral fellow. This is the things that you keep in mind. But this also gives us a lesson that until, much of the work for 30, 40 years was the normal regulation, because they were never looked outside that normal metabolism. So you always have to look outside the box and see what might be happening. And here, looking into the cancer cells open up altogether totally new way of understanding of PDC regulations and by different mechanisms, so that there is always, to think outside the box and see what can be done. And same approach can be done with designing of the small molecules. Those have been already reported. And as I have shown, when you think about it, think about something else outside the box, which will be very important in terms of, getting totally a new insight and new way of approaching the, control and metabolism, both in normal and disease states. Well, in conclusion, I want to say that, I haven't done this thing all by myself. I haven't been in the lab for a number of years. It's the people that have worked with me. I'm very fortunate to have a large number of students and postdoctoral fellows, graduate students, MS, PhD, MD, PhD students have done a marvelous job. Their dedication has really driven this field, and especially my interest in this field. And number of these students are, shown over here. It's a long list. I had trained more than 25 graduate students and 55 postdoctoral fellows over the last 50 years. So you can see that, this is their contributions. In addition to that, as I mentioned, I was trained as nutritional biochemist. I'm not a structural biologist or, molecular biologist. I have learned a good deal of molecular biology, but I have been very fortunate to have, collaborations with very, very, talented scientists. Douglas Kerr at Case Western Reserve. We did a lot of genetic defects in humans and, gene regulations. Eva Cizek has done the work with the structural aspect, crystal structure of two molecules. Frank Jordan has, been very, interested in the catalytic mechanisms, partial reactions and molecules bound to protein. And that has been very interesting to understand the catalytic mechanism of PDC. William Frey has, interacted with me and also with Dr. Jordan, and we three as a team have done this analysis of mutant molecules and this loop movement within the molecule, how the phosphorylation, interferes of disorder. And Kate Titman has done some structural aspects, and many others that I have not listed. I have a long list. So I have been very fortunate to have both, talented students and postdoctoral fellow and very active labs as a collaborator, in many parts of the world. So this has been a very, enriching experience, academic experience for me over the last 50 years. I will thank you. And here are some last people. Here is Dr. Korochikna, came from Moscow, worked with me for some 20 years as a first postdoctoral fellow, and then the research academic appointments. Dr. Malti came from Chennai, and she has done the same thing, but in the other part that I talked about it, have not talked in this particular talk is my metabolic programming, and there are a number of other students, postdoc. Here is the MD, PhD students, undergraduate students have worked. I have a long list of undergraduate students who have carried out very excellent work, and have become co-authors in many, many papers. So in short, I think it has been a blessing for me to continue on this pyruvate metabolism 5 decades with the help of talented and very committed and dedicated students and postdoctoral fellow and very active collaboration. So I thank you very much for your attention, and I look forward to listening for next 3 days as much as possible because of the time difference. But I would definitely would be very much interested and excited to follow some of the topics in the next 3 days. So thank you very much.
Thank you, sir. Thank you for a very detailed and informative session as you detailed your research journey over the last 50 years. Thank you once again, sir. That was Professor M.S. Patel, Distinguished Professor, University of Buffalo, State University of New York, USA. And now I invite Dr. Amit Patwa, Assistant Professor, School of Science, Navrachna University, to propose the vote of thanks.
Thank you, Manira Madam. I am privileged to offer a vote of thanks at this prestigious event. Gratitude is the most exquisite form of courtesy. Today, I, Dr. Amit Patwa, the organizing secretary of this virtual international conference, take this opportunity to put all my gratitude into words to one and all present here in this inaugural function. I sincerely thank our chief guest, Dr. D.S. Reddy, Director IIIM Jammu, for kindly accepting our invitation and gracing this inaugural function to address our guests and registered participants regarding drug discovery and development processes. I would like to propose a hearty vote of thanks to our keynote speaker, Professor M.S. Patel, for making the scholars understand the importance of human pyruvate dehydrogenase complexes in glucose metabolism in normal and disease states. This finding will be helpful in treatment of metabolic disease such as type 2 diabetes and obesity, as well as in some types of cancers. I also express our deep sense of gratitude to Srimati Tejalben Amin, Chairperson of Navrachna Education Society, for their support and encouragement. We have been fortunate to be assisted by our Provost, Professor Pratyush Shankar, who has provided his support and has been a constant source of encouragement to us. Thank you, sir. We also hereby render our gratitude to our registrar, Dr. Sandeep Vasan, for all the logistic and administrative help extended for the conducting this conference for the upcoming 3 days. I also thank our mentor, School of Science, Professor A.V. Ramachandran, who have been helped since the inception and shaped the structure of the conference. This function is only a trailer as we embark to the virtual journey of different aspects of organic, medicinal, and pharmaceutical chemistry. Hope to come up with a productive climax at the end of the 3-day journey that will encompass most of the disciplines related to organic, medicinal, and pharmaceutical chemistry, which will be beneficial to the mankind and the society. Finally, I would like to thank our ICT team for their support during inaugural function. Good night to all my friends in India and good day to all my counterparts in abroad. Thank you.
Thank you, Dr. Amit Patwa. To all the delegates present here, the schedule for ICOM 2022 has been shared with all participants, and we look forward to your participation, presentations, and enriching discussions over the next few days. A formal closure of this inaugural session will be with the university anthem, and may I now request all of you to please rise for the same. Thank you. We look forward to your participation over the next 3 days. Have a good day ahead.
विश्वामित्र के तट पे देखो शहर सयाजी नगरी है, माँ शारदा का मंदिर है, नवरचना यूनिवर्सिटी है। बनना है जो बन जाओ तुम, बात हमें बतलाती है, शिक्षण से ही स्वतंत्रता है, पाठ यही पढ़वाती है। नव नव रचना करते यहाँ पर छात्र और छात्राएं हैं, शिक्षण की खुशबू फैलाती नवनिर्मित शाखाएं हैं। प्रयोग से प्रेरित शिक्षा पर यहाँ जोर दिया जाता है, NUV का छात्र दशों दिशाओं में पर फैलाता है। निष्ठा तप और धैर्य यहाँ के शिक्षक की पहचाने हैं, शिक्षा और संशोधन से जो उन्नत राष्ट्र बनाते हैं। नवरचना नवरचना यूनिवर्सिटी, नवरचना नवरचना यूनिवर्सिटी, नवरचना नवरचना यूनिवर्सिटी, हमारी नवरचना नवरचना यूनिवर्सिटी।
Good evening to all the delegates. As Dr. Mandira Sikdar has mentioned, the link for the conference technical sessions has already been shared with you. Tomorrow we begin with the session wherein Professor J.S. Yadav shall be delivering the plenary talk on opportunities and challenges in drug discovery, followed by 3 other talks, and tomorrow's sessions are going to be delivered by pharmaceutical industry professionals also post lunch, and also the 3rd session will have Professor Fernando Estrada, Professor Venu Raman from John Hopkins School of Medicine, and Dr. T. Rajamannar from Sun Pharma Advanced Research Center. We look forward to the presence of all the delegates tomorrow. Thank you. Thank you very much.
Thank you all. My special thanks to Professor Ramchandran for invitation. It has been very enjoyable beginning, and I look forward to listening to several presentations over the next 3 days.
Thank you. Thank you very much.
Thank you, sir.
Thank you.
Good night to all from India and a good day to all from the North American continents.
Thank you.
Thank you. Thank you.